Welcome back. This is now the second part on heredity, which is actually a brief discussion on human genetics. And human heredity, the study of the transfer of um, hereditary traits among humans, came way back about 100 years after the Mendelian genetics. We started pretty much like before that, but we were not successful in establishing a lot of uh, paradigms on human heredity because technology was not yet so advanced. Now that we have already discovered the different genes, the location of the genes, the chromosomes, we got in depth into the human genome, and heredity, or the study of the transfer of traits among humans, are now full blast. So let's take a look at some of the things that pretty much you are going to be tested on in human heredity. First of all, the human chromosomes. The human chromosome would number 46. There's about 23 pairs. 23 out of the 46, 23 is coming from the mother, and then the other half, which is another 23, is coming from the father. Cell biologists would analyze the chromosomes by looking at those paired up chromosomes, and the picture, I call it the class picture of the human chromosome, is actually the karyotype. The karyotype can actually give you a lot of information. For instance, it'll tell you if the chromosomes are complete, because we have 46. It'll tell you the sex of the bearer of this particular chromosomal set. And if you look at the sex chromosome, and that's right about here, you have the X chromosome, which is actually very big, and the Y chromosome, which is really small. This is actually the smallest of all the chromosomes. You say that this is really a male. If you have two X chromosomes right there as a number 23 chromosome, or the sex chromosome, then you have a female. So you could see also in a karyotype if there's going to be an extra chromosome in any of the sets. All right. You can also see if there's something wrong physically with a chromosome. Is there going to be an inversion? Is there going to be like a deletion? Because these chromosomes are homologous. And you know what homologous pairs are? They're supposed to be bearing the same traits on their bodies, and therefore they should have the same bands and they should look alike. Right? The only thing that's going to be different with them is some in what you call this, in some of the traits, one of you will probably have the dominant trait, the other one would have the recessive trait, or a heterozygous or homozygous. So the the traits, the forms of the alleles are the only things that's different, but all of the alleles that are present in each of the 23 chromosomes, they are pretty much the same. So the cells are photographed here. How do they do this? How do they collect these chromosomes? The cells are photographed during mitosis. Scientists would cut the chromosomes from the photograph and group them together in pairs. And they're actually separated when they are already in metaphase. So that when they are in metaphase, it's easier to pretty much like put them together. So a picture of these chromosomes, the whole set itself, is what you call a karyotype. Here these are an odd pair. Okay, this is a supposedly a homologous chromosome, but this is the only pair in the set of 23 pairs that we have that is not necessarily a pair, most especially among men. This is actually the sex chromosome. Two of the 46 human chromosome, known as the sex chromosome, is not really homologous. Females, though, in a female, because we do have two of the X chromosome, this is actually an X chromosome, then those our X chromosomes are homologous. But the males have one X and one Y, and they are not homologous. And the remaining 44 chromosomes are known as autosomal chromosomes or autosomes. So these are this pair right here is the sex chromosome, and the rest of them that is not X, that is neither X or Y, will be your autosomes. So which chromosome among the 46 would determine the sex of a child? How is sex determined? Well, all human egg cells carry a single X chromosome. That is the egg cells. Okay. And so the number 23 chromosome is actually an X chromosome. That's actually in the egg cell. And then in the sperm cell, half of the sperm cell carry an X chromosome, and half of the sperm cells that they produce would carry the Y chromosome. So about half of the zygotes that will be the result of fertilization, the probability of a sex will be 50-50. 50% probability for being a male and 50% probability for being a female. So right here you have a female, and a female egg always have two X chromosomes, okay, or rather has an X chromosome, and a female egg always has an X chromosome because we do not carry any Y chromosome. But the male have both X and Y in their what do you call this, in their spermatogenesis, and pretty much when these develop, they will be developing 50% of them will be like sperms that carry the Y chromosome and sperms that carry the X chromosome. When either one of them, the X and the Y sperm, will fertilize an egg, the resulting would either be an XX. If an X sperm fertilizes an egg, it will become an XX. And that's about two of them out of four possible gametes and four possible zygotes. And then if the sperm is a Y, and then it fertilizes the egg, and the egg is still an X. Can't change that anymore. But the Y will fertilize an egg and it'll become an XY, which is a male. And out of the four possible zygotes, you have actually two that will be males. So 50-50, 50% chance. And who would decide on the sex? Well, there's really no true decision, but it's actually the Y chromosome. The Y chromosome dictates who's going to be male. If you have the Y chromosome, you are a male. If you do not have the Y chromosome, you are a female. Whether or not you have only one X abnormally or two X's normally, you are going to be a female because you do not have the Y chromosome. But the moment you have the Y chromosome, regardless of how many X chromosomes you have, if you have an XY, that's a normal male. If you have an XXY, that is a Klinefelter, an abnormal, but still a male. So human traits pretty much would also um, apply the Mendelian genetics. Biologists must identify the inherited traits that is controlled by a single gene, because Mendelian genetics pretty much explains only the principle of dominance and principle of segregations and independent what do you call this, independent assortment. Then they really do explain only the simplistic heredity, human heredity. They must establish that the trait is inherited, and pretty much if you're going to use or apply Mendelian genetics, they'll have to establish that the genes or the hereditary trait is not a result of environmental influences. They have to study how the trait is passed on from one generation to the next, which is pretty much not anymore a magic trick. We know already how they are being passed on. But the hard part, the difference between the Mendelian study and the human genetics, a study of human genetics, is actually the number of years by which we will need to wait for an offspring really to pretty much like pop out and explain to us the result of the mixture of genetic traits. For the piece, Gregor Mendel pretty much did not wait long, and in a matter of days or months, a few weeks, he already knows the answer to his questions. But with us, it'll take decades before we can actually see the results of the what do you call this the cross in humans. Plus, there is also the question of ethics. Okay, we can just cross peas, they're peas, but we cannot just cross humans just because of an experiment. There's actually the question of ethics there. So, how do we study? How do we study um, the transfer of genetic traits? We go back. We go backwards. Instead of going forward, we go backwards. We learn the transfer of traits backwards. We're going to look at our own traits, and then we're going to look at from whom among our parents we got those traits, and from whom among your parents got them from their grandparents, up to as far as we can in terms of information, in terms of information, as far as we can reach. And that's actually what you call a pedigree chart. Genetic counselors analyze pedigree charts to infer the genotypes of family members and how these genotypes transfer off to the next generation, or how it was transferred off to the next generation. Because pedigree charts are results of transfer that has already happened. You're going backwards. You're not going forward. But then by looking at the pedigree charts, you can actually determine what the future may hold in terms of transferring the traits in the future. So let's take a look at how you can read the pedigree chart. A square in a pedigree chart would mean a male. And of course, your circle would represent a female. Now, the lines that are connecting the vertical line and the bracket that will actually be connecting the parents to their children. Okay, and the vertical line connects to the children, and all of these children, the horizontal lines are all connected to each other, and that means they are siblings. And this is actually, these two are their parents. A circle or a square that does not have the shade, the color, it's not colored, it's just an empty circle or square. It indicates that the person does not express the trait that we are studying. And then you have a horizontal line connecting a male and a female, that means a marriage. And a shaded circle or a shaded square indicates that the person would express the trait that is being studied. Pretty much these are the only things so that you will know exactly how to read a pedigree. You might be given a pedigree in your exam, and I am pretty sure I'm going to give you a pedigree in your worksheet so you will know and you will understand, and you can actually study in depth how to understand a pedigree. So let's take a look at the genes and the environment. There are some obvious human traits that are almost impossible to associate with single genes because most of our traits are actually polygenic. Traits such as the color, the shape of our eyes, the color of our eyes, the shape of our ears are really polygenic. I cannot even think of a single trait in our body, in our human body, that are expressed because of a single gene. Our traits are controlled by many genes, and many of our personal traits are only partly governed by genetics. Some of them are controlled by the environment. Like for instance, the color of our skin is a is actually a very classic example of a trait that is controlled by our environment. Color of our skin will change because of our exposure to the environment. The sun darkens the skin. The weather will actually make the skin look different. Okay, uh, so the environment pretty much has a lot of things to do with how our genes are expressed, our traits are expressed. The human genome includes tens and thousands of genes, about 35,000. And each of these genes interacts with one another and therefore can express traits that is more than 30,000. Say, how can the human genes be only like 35,000 when we have more than 100,000 traits? Okay, our traits is not limited only to the color of our eyes or color of our hair. It's the traits also has got something to do with the type of protein that you express. Are you lactose intolerant? That's being a trait. Can you are you able to produce a specific type of protein? And is this protein able to interact with another protein? Those are actually traits. Even the interaction is a type of trait. And therefore, you can actually count more than 100,000. The proteins alone in the body would pretty much like count more than 100,000. So, how can 35,000 genes account for more than 35,000 traits? Well, it's actually the interaction of genes. When one gene or two genes would interact, they form a specific trait. When gene A, B, C interact, they form D. When gene A and B interact, they form F. When gene A and D interact, they form X. So the interaction of an X number of genes will actually express a trait or several traits. Plus the pattern of interaction and what you call is the completeness or the incompleteness of interaction will also pretty much like express different genes. The human the human genome so includes uh, more than 10,000 genes, approximately 35,000. And in 2003, the DNA sequence of the human genome was published and was uh, based on 5 individuals of different race, 5 individuals only. And now I believe they have more than 5 individuals, and those were only the first 5 that pretty much like agreed to so that we can use them or we can use their genes to study the human genome or to establish the human genome. Now, in a few cases, biologists were able to identify genes that directly control a single human trait, for instance, such as a blood type. So, the blood group genes. The human blood comes in a variety of genetically determined blood groups, and a number of genes are responsible for the human blood groups. The best known in education is usually, and well, because it's well studied, is actually the ABO system and the RH blood groups. The RH blood group is determined by a single gene with 2 alleles. That means being positive and being negative. Being positive allele is dominant, so individuals who are Rh positive are said to be dominant over the Rh negative. So the Rh positive means you have uh, the protein, the R, the R, the RHO antigen. Having the RHO antigen is actually, or having the chance or having the ability to produce the RHO antigen makes you an Rh positive. You could be homozygous for being Rh positive or having the Rh antigen, or you could be heterozygous. Or you probably do not have the ability at all to produce the Rh antigen, in which case you are an Rh negative. Now, individuals with two Rh negative alleles are really completely Rh negative, but an Rh negative paired up with an Rh positive allele would be an Rh positive, heterozygous Rh positive. Now, the ABO blood group would simply mean blood type A, B, and blood type O. And pretty much the three alleles for this gene is your allele to produce the antigen A, the allele to produce the antigen B, and the allele not to be producing the A or the B. So allele A and B are both dominants. When the individual is capable of producing both A and B, then both of them will be expressed. You will have AB. Okay, nothing will be masked off because both A and B are codominant. Now, here is actually your blood type. AB here, blood type AB would have the genotype. They carry the genotype A and then the genotype B, and that's an AB. And because they're codominant, then you express it as AB. Now, you can safely transfuse AB to blood type A, B, AB, and O. AB is a universal recipient, and you'll have to remember that. Now, the I in the allele of the ABO system means recessive. The I means inability to produce. Okay, so let's say your genotypes for blood type A. Blood type A, you have an individual here which is homozygous for blood type A. You have, because he got his blood type or his ability to produce A, one from his parents, both of his parents gave him the ability to produce A. So he's blood type A, homozygous A. The other genotype is IAI, which means one of his parents gave him the ability to produce A. Right there is the ability to produce A. But his other parent gave him the inability to produce A. And because the ability to produce A is dominant, then you are expressing the A. You're still blood type A, even if only one of your parents gave you the blood type A. Now, the antigen that is actually on the red blood cell of blood type A is the A antigen because you are able to produce that antigen on your red blood cell. That's why it is blood type A. Now, blood type A is safe to transform to blood type A and is also uh, it is also safe to transfuse to blood type. I don't know about AB. Yeah, AB, because AB is a universal recipient. However, you are actually able to receive only, if you're blood type A, you're only able to receive from blood type A and blood type O. As a matter of fact, blood type O is the universal donor. Now, individuals who are blood type B would have the genotype IBIB, where both of the parents actually gave him the ability to produce B, or if only one of the parents gave him the ability to produce B, the other didn't, it would still be a heterozygous, and because B is dominant, he will still be producing the blood type B and will be expressed as blood type B. Individuals who cannot produce either blood type in the antigen A or the antigen B would have the I allele. And this I allele, if you are unable to produce either A or B, would be a double I. And a double I allele would signify blood type O. And there's no antigen on the red blood cell, and therefore you can give blood type O to any blood type because of the absence of antigen on the red blood cell. It is safe to receive blood type O even if your blood type A, B, AB, or O, because it's a universal donor. So, recessive alleles. The presence of a normal functioning gene is revealed only when an abnormal or non-functioning allele would affect the phenotype. And therefore, many disorders are actually caused by recessive, autosomal recessive allele. I'm not even saying sex chromosome, okay? Autosomal recessive allele. So, an example of these recessive alleles will be your albinism, or the lack of pigment in the skin and the hair. Okay, if you have two of the recessive alleles, then you're going to be an albino. Cystic fibrosis, which is accumulation of excess mucus in the lungs, in the digestive tract, in the liver, and increased susceptibility to infection, pretty much is because of the absence of what we call this, the correct CFTR gene. In what the cystic fibrotic people have will be um, a faulty CFTR gene, and that's already, it's only like um, very small, it's only a very small fault, but yet it will already be a syndrome. It will already affect a slew of metabolic reactions in the body, and it could actually shorten the lifespan of people with cystic fibrosis. Galactosemia is the accumulation of galactose, which is sugar in milk, in tissues, and a cause, and then the result of this will be mental retardation and eye and liver damage. Then you have PKU, or phenyl ketonuria, which is an accumulation of phenylalanine in the tissues, and the lack of normal skin pigment and mental retardation. These are all recessive alleles, all right? And then, of course, your Tay-Sachs disease, which is accumulation of uh, lipid in the brain cells. And this can result to mental deficiency, blindness, and even death in early childhood. These are all disorders caused by recessive alleles. You need to have significantly two forms of the same recessiveness so that you can actually, so that you will present the phenotype of these disorders. But the disorders that are actually dominant in nature are more critical. The dominant alleles, the effect of dominant alleles is actually its full expression, even if the recessive allele is present. So even if you are a heterozygous for that particular trait, the, the expression is going to be there. And two examples of genetic disorders that are caused by autosomal dominant alleles are achondroplasia and Huntington's disease. Achondroplasia, I'm pretty sure you are familiar with achondroplasia. Achondroplasia is actually dwarfism. Remember those dwarfs? They are pretty much like intelligent. They are, they're just small, and they have short limbs, but they are pretty much normal in other ways except their size. So you need this disorder is caused by a dominant allele, and two forms of the dominant allele for achondroplasia would pretty much like result in a dead fetus. So the fetus is not going to survive. It is, if it is actually a double A dominant. Okay. So for achondroplasia to happen, it should be a double A and then a recessive. All normal sized people would actually be recessive for achondroplasia. If we don't carry it, then we are small r, small r. If we have a big, let's say A is actually achondroplasia, not R, A. If we have a, small a, then that's recessive. We are normal. If we have a big A and small a, then we have achondroplasia. Okay, and so a live achondroplasia. If you have a double A, capital, uppercase, meaning dominant trait, then that means that fetus is not going to survive. So, Huntington's disease. Huntington's disease is a disorder that results in mental deterioration and uncontrollable movements, uh, usually that appear in the middle age. So you would not know if you have Huntington's disease unless you are around 40, 45 years old. And sometimes it is too late for a lot of middle-aged people. Mostly at that age, we already have our family and we already have our children. And for the most part, these particular disorders are actually genetics, and therefore you could hand this down to your offspring. And too bad, no matter how much you would want not to affect your child, you might have already affected your child if you are carrying the Huntington's disease. If you are carrying the Huntington's disease and it's a dominant trait, whether you are a dominant or heterozygous or homozygous, you are going to pretty much like go through the process, the painful process of until death, effects of the Huntington's disease. Then you have hypercholesterolemia. We all know from, and you will too, we discover it that when you get into your digestive system and the effects, or rather the actions of your liver, you will know that the liver really naturally produces cholesterol, because we need cholesterol in the production of our cholesterol-based hormones, estrogen, progesterone, testosterone, hydrocortisone. These are all cholesterol based. So we naturally produce enough cholesterol, as a matter of fact, sometimes too much cholesterol, just so we can have these hormones. Now, if you eat more than what you should be eating, more than what you can actually like break down, if you eat cholesterol loaded food more than you can break down, then you're dangerously like increasing your the amount of your cholesterol, and the chances of that being deposited in your arteries will be bigger. Excess cholesterol in blood would eventually cause heart disease. And if you have hypercholesterolemia, that means you are actually producing excessive amount of cholesterol, much more than the normal. And this is actually caused by a dominant allele. Codominant alleles among humans are, uh, well, the ones that are actually very well known is sickle cell anemia. And the ABO system is also codominant. Both A and B are dominant. In the sickle cell, okay, it is a serious disorder that is caused by a codominant allele. The sickle cell is found in about 1 out of 500 African Americans. And the, the disorder, sickle cell, would have a major symptom that affects the red blood cells. It will cause a sickled shaped red blood cell, and it can cause what it calls damage to many tissues because of the inability of the blood to to carry oxygen because of the sickle shaped red blood cells. So, how do small changes in the DNA cause a genetic disorder? Well, for instance, both cystic fibrosis and sickle cell anemia disease had only like one nucleotide change. It's only a single nucleotide, point mutation in the gene itself, and it affects already the whole entire structure of the protein, and it can cause already a serious genetic disorder, and it pretty much like becomes a syndrome. So cystic fibrosis is caused by a recessive allele, and sufferers of cystic fibrosis produce a very thick, heavy mucus that clogs their lungs and breathing passageways. And the most common allele that causes cystic fibrosis is missing the 3 DNA bases. And if you miss the 3 DNA bases in your CFTR, CFTR stands for cystic fibrosis transmembrane protein gene. Okay, as a result, the amino acid phenylalanine is missing from the CFTR protein. The sickle cell anemia is caused only by a single displaced uh, nucleotide base, and is actually replaced by an erroneous nucleotide base. And you can already have a lot of problems with it. And this is characterized by a bent, twisted shape of red blood cells, and you could see that it's not really like the usual rounded red blood cell. And this type of red blood cells would not be carrying the right amount of oxygen. Now, hemoglobin is a protein in red blood cells that carries oxygen. If your hemoglobin is unable to produce the right amount of oxygen, then you are always going to be anorexic. The sickle cell allele, okay, pretty much even if it is only one DNA base that has been changed, can create an abnormal hemoglobin that is less soluble than normal hemoglobin, and they cannot carry enough oxygen. One hemoglobin is supposed to be carrying 4 oxygen molecules. A sickle cell will be carrying only 1 or 2. So the only thing that you could do with a person who is expressing the phenotype of sickle cell anemia will be to give him every 3 months blood transfusion. That's all, because he is not producing any good red blood cell. So people who are heterozygous for the sickle cell are generally healthy. They are carriers. They are resistant to malaria. But it is important to note that while the sickle cell carrier provides some protection against malaria, it does not eliminate the risk of infection. Individuals with sickle cell trait can still contract malaria, but they are generally less severely affected. And there are 3 phenotypes that is associated with the sickle cell gene. An individual with both normal and sickle cell allele has a different phenotype, is resistant to malaria from someone with only 2 normal alleles. The sickle cell alleles are thought to be, both of them are dominant, so they are codominant. And here you could see the distribution of sickle cell allele and malaria. Okay, this is actually the region in the world where malaria is common. And then this is actually the region in the world where sickle cell allele is common. Color blindness. I'm pretty sure color blindness is one of the things that you are going to pretty much like have in your final exam. There's the 3 human genes that is associated with color vision, and they are located in the X chromosome. So if you are colorblind, whether you are male or female, okay, just know that it has been transferred to you by your mother. In males, a defective vision of any of these genes would produce color blindness. And it's because it is a recessive gene and you find it in the X chromosome. The expression of this gene is most likely among males, because males have only one X chromosome. So if they carry the recessive gene, they do not have another X chromosome that can carry the normal dominant gene that could mask the effect of this recessive disorder. Obviously, they are going to have it. Once they have it, they are going to be colorblind. Okay, so make sure that you know. For instance, here is actually where your father is a normal vision. This is actually the father itself, and the father is not colorblind. Okay, then their son would have normal vision. Okay, but the mother here is colorblind. You can see this X chromosome here that would carry the recessive trait. They have a daughter that would carry the trait, but is a normal vision, but she's a carrier. And then this X chromosome is going to be handed down to the son, and it's going to make the son colorblind. So if you are colorblind, gentlemen, do not blame your father just because you both have the Y chromosome. You're colorblind because your mother handed down the color blindness to you. Hemophilia is the same thing. It's also an X-linked chromosome. It's an X-linked gene, pretty much that help control blood clotting. And if you do not have, and hemophilia will, this recessive gene pretty much like renders a person unable to produce the blood clotting factor number 8, which is the anti-hemophilic factor. It is a recessive allele, and in any of in any of the two genes that you have, it may produce hemophilia. And uh, the protein that is necessary for blood clotting, the factor 8, is missing in hemophilia. And the hemophiliacs can bleed to death from cuts and may suffer internal bleeding if they are bruised. Muscular dystrophy. Duchenne muscular dystrophy is a sex-linked disorder that results in the weakening and loss of skeletal muscle. It is caused by a defective version of the gene that codes for a muscle protein dystrophin. Then you have also the X chromosome inactivation. Ooh, you could just imagine how much gene and how much phenotypes and genotypes is actually carried by the X chromosome. It is big. It has a lot of genes. Now, a British geneticist, Mary Lyon, discovered that in female cells, one X chromosome is randomly switched off, and the one that is sometimes uh, between the two X chromosome in a female, certain aspects of the X chromosome, the pair, can actually be switched off. When one aspect is switched off on one, and the other one is switched on. That's why we have actually traits that can actually prove the switching on and off of a particular gene. And uh, an example of this is actually and among animals will be your calico cats. The calico cats having that spotted brown and black on over white is actually the X chromosome that carries X black in brown, rather brown in the X chromosome and black in the X chromosome. Both of these are actually like both of these colors are actually like dominant, and there are some parts of the X can actually be activated and will form and will show brown coloring on the fur of the cat, and then some part of the other X can express the black and can form the black spots on the fur. But the white, which is really what the color is actually over on, is actually recessive. But you could see the white, but you could see specks of either black or brown, which is supposed to be codominant, that is actually actively like actively, you can actively see that over white. And this chromosome forms a dense region in the nucleus, which you call the Barr body. Barr body is actually the chromosome that is actually like shut down. Okay, they are actually like clustered together, and they are not expressed. Barr bodies are generally not found in males because their single chromosome is they have only one X chromosome, and therefore there's no way of switching off the X chromosome in males. They're always switched on because they have only one X. So let's take a look at chromosomal disorders. Disorders that is brought about by chromosomes. What problems could that be? Well, sometimes whenever cells divide and the chromosomes will undergo and then the cells will undergo meiosis, there are some problems with how the chromosomes will behave in meiosis, and it will cause what you call non-disjunction. I have uh, I have the notion that you are going to deeply engage in learning this when you get into the reproductive system, when you study oogenesis and spermatogenesis in the reproductive system. However, I'm going to give you a little bit of what this non-disjunction means. This is the most common error in meiosis that occurs when homologous chromosomes during meiosis will fail to separate. And it is known as non-disjunction, which means they are not coming apart. And here is actually a diagram of meiosis. If non-disjunction would occur, the abnormal number of chromosomes may find their way as becoming part of the gamete. Let's take a look at that. This chromosome here, which has an original chromosome number of 4, okay, will form gametes. And the gametes are usually haploid. They carry 1/2 the number of chromosomes from the mother cell, all right? And you can see that these are supposed to be all haploids. Now, during the first meiotic division, what happens is that the chromosomes, the homologous chromosomes, will have to separate. Okay, there's not going to be any division along the centromere, but the homologous chromosomes are just going to separate. They will say goodbye to their pair partner. And that is after they undergo crossing over. And crossing over means an exchange of genetic material. So after they're able to exchange genetic material, they say goodbye to one another. And they have done already the first meiotic division. Sometimes, take a look at this, guys, that particular couple pair of yellow homologous chromosome, they did not separate. Only the pink separated, right? These two, they were so in love with one another, they don't want to separate. So they went into the same chromosome, into the same cell. And so this cell now will divide via their centromere to give rise to the haploids, to the haploid cells. And here you have the haploid cells. You're supposed to have 2 because the original number is 4. So the haploid number is 2. And this has only 1. 1. You're supposed to have 2 because the original number is 4. And these have 3. 1, 2, 3. Why? Because those guys there did not separate. So here, this is what you call an N. An N is supposed to be a haploid. 2N is supposed to be a diploid. But this is an N plus 1. This is an N minus 1. These are all gametes. Okay, they could be egg, they could be sperm. When they fertilize their counterparts, these abnormal gametes here will have 1 chromosome more than the normal 46. This will have 1 chromosome less than the normal 46. And you could just imagine this is only because of meiosis. And meiosis is happening in the production of gametes. Whether you're talking about egg or sperm, both gametes undergo meiosis. If they will have non-disjunction in meiosis, they will be producing either an N plus 1 gamete or N minus 1 gamete. Now, if the non-disjunction happens in meiosis 1, all of the daughter cells are going to be abnormal. If the meiosis 1 division, they were normal, and then non-disjunction happened in meiosis 2, then only 50% of their daughter cells will be defective. Down syndrome is a human disorder where 2 copies of an autosomal chromosome fail to separate during meiosis. This is a classic case of non-disjunction. An individual may now be born with 3 copies of a chromosome, an N plus 1, and is actually fertilized by a normal gamete, will now be producing, instead of 46 chromosome, the person will have 47 chromosome. In this case, the Down syndrome person will involve 3 copies of the chromosome number 21. And that is your chromosome number 21. One of these 3 guys here did not separate with their homologous chromosome, and they became what do you call this? A triple. All right. Down syndrome produces mild to severe mental retardation. This karyotype pretty much causes mental retardation and various physical problems. People with Down syndrome can, however, lead an active, happy lives, pretty much, but uh, they have some issues. They are susceptible to many diseases and a higher frequency of some birth defects. So there are also sex chromosomal disorders in females, and non-disjunction of the X chromosome can lead to Turner syndrome. A female with Turner syndrome will have only one copy of the X chromosome. So this is an N-1, and the other X chromosome came from the normal, and there's one of the gametes did not have any X chromosome because of non-disjunction. So the chromosome number of Turner syndrome people will only be 45 instead of 46 because they're missing one X chromosome. Women with Turner syndrome are sterile. In males, there's also non-disjunction of the X chromosome that can produce a double X and a Y. Remember, if you have a Y, even if you have a double X, you are not going to be a female here, you are going to be a male. So your total chromosome number here is 47. The extra X chromosome interferes with meiosis and usually prevents this individual from reproducing. Okay, let's take a look at some. Oh, I see already that we already finished with the information. We are now going to get into a mock quiz. The average human gene consists of how many pairs of DNA? Well, pairs of DNA average is about 3,000 pairs of DNA. Which of the following genotypes indicate an individual who is a carrier of color blindness? An individual, a carrier, well, I'd say a carrier will be a female, so it's only between these two, right? This is normal. I think this is a carrier. This is a male that is colorblind, and this is a male that is normal. All right, we're right. That is a carrier. Color blindness is much more common in males than in females because the recessive gene on the male single X chromosome is expressed. I think I don't need to read anyone. I think that's the answer right there. That's the first one. Absolutely correct. The presence of a dense region in the nucleus of a cell can be used to determine pretty much, I say, the sex of an individual. Okay, that's actually the Barr body. Only females will have Barr bodies. Now, non-disjunction that occurs during the stage of meiosis will produce 100% non-viable gamete will be meiosis. I just mentioned this to you, meiosis 1 only, 100% non-viable gamete. Then a chromosome that is not a sex chromosome is known as an autosome. Excuse me. Whether a human will be a male or a female is determined by, I think, a sex chromosome is in the sperm. Whoa, yes. In the Mendelian inheritance in humans, they are usually studied by, well, we cannot do controlled process because of ethical reasons. I think we only make inferences from family pedigrees. Yes, very good. An individual with blood type O phenotype can receive blood from an individual with a phenotype. I think O, because O can only be transferred to an O. Good. The ABO blood group is made up of how many alleles? Well, A, B, and the allele for the O also. All right, three alleles. That wraps up our genetics. And I will see you in the final exam. Good luck, everyone.